that we have the tech worked out. I hope so. Hi everyone. I am Sarah Smith in the Biological Oceanography Lab, and I'm happy to welcome you to the defense of Hannah McGrath, who is the first student to graduate from the Bio-Ocean Lab since I joined Moss Landing a few years ago as her official advisor. I've had the pleasure of working with Hannah over the past 3 years and having her in class. She's an excellent student and has been really motivated throughout this process to finish her degree quickly. And I'm really excited to hear more about her research project that was supervised by her primary advisor, Dr. Holly Bowers, in the Environmental Biotechnology Lab. So I'm here in an official capacity, and we'll just let you all know that over the next period of time, you'll be hearing from Hannah about her thesis research after an introduction from Holly. And then her thesis committee will adjourn for a period of time at around 11 o'clock after the defense to discuss how things went. And after that period of time, for those of you who are in the audience, to stay and join Hannah afterwards for hopefully some congratulations. So I will, without further ado, just turn it over to Holly to give the formal introduction. Thank you.
As Sarah was saying, Hannah came here a mere 3 years ago, and she's already, I think she had her thesis proposal on my desk in like a month or something like that. She showed up with ideas, and so we got to work. And so in the spirit of Hannah, if you know her, she loves lists, loves to-do lists, loves checking things off. If you ever meet her in the hallway and say, let's have a meeting later today, you turn around, there's a Google invite. So very, very organized. So in the spirit of that, we are going to go through a checklist. Let's see if I can get this to work. So here's our checklist. Who is Hannah McGrath? Hannah joins MLML. I don't know why they're all ones. They were 1 through 5. Obviously, I didn't learn anything from you when you were in the lab. We'll talk about Hannah working hard, Hannah going to Japan, and Hannah having fun. So who is Hannah? She came to us from New York City, for those of you who didn't know. So, it's an obvious transition to say, I live in New York City and I'm going to go to Moss Landing. I mean, there's a stark difference. I remember her coming here and driving for the first time. I was like, if you learned in New York City, I mean, you got one road between here and Marina, you can't mess it up, and it's easy. She got her BA at Colby College in Maine in 2022 with a major in environmental science. Must be a glutton for punishment because she was doing managerial economics on the side for a minor. And she did a semester course at Bigelow in Maine, as well as an NSF REU, which are highly competitive, at the Mote Marine Laboratory in Florida. And that's where she got her taste for halves and reached out to me. So check, we got that, we know who Hannah is. Okay. Next, Hannah joins MLML. So she dove right into classes. I think she was, prepared on day one to show up. She did try to bring a bit of her New York City thug life with her, but it didn't take hold here at MLML. So she quickly transferred into her nice, sweet, innocent little self. So she joins MLML. Here she is. Hannah works hard, so if you've ever seen her in the lab, she's always in the lab, sun up, sun down, and did some of her work as well here up here on the hill in the Bio-OSH lab. So on the left, she was at the microscope a lot during these 3 years because she ran our Monterey Wharf weekly monitoring for phytoplankton program, so she was always there. She also got drugged literally into the field a few times, so she'll do anything. Just, she's too nice, she can't say no. So crawl in the mud, yep, you got to do that. So we know she works hard. Hannah goes to Japan. So a couple of years ago, EBL got the opportunity to go to the International HAB meeting, and both her and Olivia raised every single penny for their travel, which was pretty impressive. And so Hannah did an outline, a poster, and I believe a speed talk too about our phytoplankton monitoring, and so that was nice to put sort of on the world stage because that's part of Cal HabMap, and Rafe Kudella's here, and their lab does the Santa Cruz Wharf. So it's a whole Cal HabMap program, so that was nice to introduce that to on the international stage a bit. And somehow on banquet night, she ends up on the stage with a sword and some dancing dragons. I'm not quite sure how that happened, but that was the last night. And later that week, they ended up in kimonos as they were traveling around. So Hannah got to go to Japan. Hannah has fun. I was happy to see some fun pictures when I was going through all of this, not just all work, 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 work. So Tanya, who's here in the back, her and her family did a little movie about EBL and what all of our projects were. It was a lot of fun, but we all had to wear makeup. Like, she put lipstick on me. It was kind of strange. And then her and Olivia were usually up to something. There they are in my very outdated kitchen, planning and scheming something. And then she has even more fun. So it's nice to see you out and about and relaxing and having a glass of probably water, but so we know Hannah has fun. So with that, I will end here. It has been such a pleasure to have Hannah in the lab because she's just been really helpful with everything. The dishes need washing, she does them. Experiments need to be done, she does it. It doesn't matter what it is. And so I am more than happy to turn it over to her to let you hear about all of her fun work over the past mere 3 years. So thank you.
Thank you, Holly, for that lovely introduction, very sweet introduction. Is it this one? No, I don't think so. Hold. Okay. Yes, thank you, Holly, for the lovely introduction. It's been a pleasure, and I want to thank everyone here today joining me in person or via Zoom. For my thesis defense, and I'm thrilled to share my thesis work titled Temperature Effects on Growth, Community Composition, and Domoic Acid Production in Pseudo-nitzschia Species. And this research was made possible with Dr. Holly Bowers and my committee members, Dr. Sarah Smith, Dr. Thomas Conley, and Dr. Rafe Kudella, who are all here today. It is. Okay, so we're going to dive right into it. Sea surface temperatures in the California current system are increasing. So the image to the right shows the California current system. Meanwhile, the figure to the left has year on the x-axis and temperature on the y-axis. So those red oscillating lines represent recorded sea surface temperatures, and the blue lines represent predicted sea surface temperatures from a model. So the horizontal lines represent mean sea surface temperatures, and Shiyu et al. 2018 in this figure predicts that by 2030 to 2049, sea surface temperatures are expected to increase to about 17.1 degrees Celsius, and this is represented by that pink horizontal line. Similarly, if we zoom into the California current system, into Monterey Bay, which is where we are at the moment, Monterey Bay here, there is a long-term trend for the Hopkins Marine Station showing that from 1920 to 2020, sea surface temperatures have increased, which is represented by those positive slope lines on the graph. So now that we know sea surface temperatures are increasing, why do we care? How does this impact us? So rising sea surface temperatures are associated with many consequences to the ocean ecosystem, like changes to marine food webs. For example, organisms may shift northward to colder waters, decreased oxygen levels, rising sea levels, and intensifying harmful algal blooms. And this is what I will be focusing on. So what are harmful algal blooms, or HABs? They are species of phytoplankton that produce harmful toxins or are nuisances to the environment. And to the left are common HAB species found here in Monterey Bay. So we have Dinophysis, Pseudo-nitzschia, Akashiwo, and Alexandrium. They all have their different characteristics and look, I think they look charming. But there are various factors that support these blooms, and bloom events like nutrient inputs, rainfall, salinity, wind, temperature, pH, and light. Although phytoplankton cannot be seen with the naked eye, they can have a pretty disproportionate effect on the environment. And so for my thesis, I studied the diatom genus Pseudo-nitzschia, and it kind of looks like a needle under the microscope, so this was taken by the Cadella lab. And there are more than 52 species of Pseudo-nitzschia that have been recorded, and of those, half produce a neurotoxin called domoic acid, or DA, which I'll be referring to further as we progress in this talk. So Pseudo-nitzschia was first recorded to produce DA in 1987 on an island called Prince Edward Island in Canada. And since it has been recorded worldwide, which you can see was reviewed by Bates et al. In 2018, so all of those little markers indicate a place where a different species has been recorded. So it's worldwide. So what is DA? It is a neurotoxin, and it binds to glutamate receptors, and this can cause swelling and death of neurons. So here's a diagram of how DA is transferred into the food web. So the toxin is transferred into the food web. First, we are exposed as humans and marine mammals through the ingestion of either finfish or bivalves or tasty crustaceans, but when they're contaminated, we can get poisoned. And so when humans are poisoned, it's called amnesic shellfish poisoning, or ASP. And when marine mammals like sea lions or birds get poisoned, it's called domoic acid poisoning, or DAP. So symptoms can include short-term memory loss, seizures, and in extreme cases, death. So this diatom did gain major attention in 1963 when the horror movie, I don't know if any of you have seen it, but it's a classic, The Birds. It was directed by Alfred Hitchcock, and this movie was actually inspired by a DA event pretty locally in Capitola when birds started attacking people. And so this is the flyer they used to promote the movie. So this diatom also gained attention in the scientific community. So in Monterey Bay in the fall of 2013, a pretty diverse toxic bloom, but it was primarily dominated by Fraudulenta, reached abundances as high as 1 million cells per liter. And the darker and larger circles indicate higher domoic acid concentrations or levels. And this was primarily detected offshore in Monterey Bay. But then in 2015, warmer waters than usual, coined as the warm blob, occurred, and this supported a record-breaking toxic Pseudo-nitzschia bloom that was dominated by a different species called Australis. So this bloom stretched all the way from Southern California all the way up to Alaska. So it had some pretty devastating effects. And as you can see, those larger circles in the 2015 panel are larger and darker, indicating that it was near shore and more intense than the 2013 bloom. So as a result of the warm blob intensifying the Pseudo-nitzschia bloom, marine life did suffer. So I consider them as losers. So we have crab seeds, so the Dungeness crab fishery lost over 48 million due to closures, seabird mortalities, as well as marine mammal mortalities. And this image was actually taken on the Mbari beach, so right behind us, pretty local. So the warm anomaly alongside the massive DA event sparked many studies like these. They just kept coming out. Everything was about the warm blob and harmful algae blooms. And I've highlighted diversity in red because few papers have actually investigated the impact of temperature on Pseudo-nitzschia diversity. In fact, it hasn't yet been carefully evaluated experimentally, representing a knowledge gap that needs to be addressed to understand the impact of increasing sea surface temperatures on Pseudo-nitzschia community composition. So as I previously mentioned, there are more than 52 species of Pseudo-nitzschia, and half of those produce domoic acid, and it's important to identify which ones are toxic. So in this background image, which is an image under the microscope, how many species of Pseudo-nitzschia can you identify? Anyone want to take a stab? Remember, they are the needle-like ones. 4? Anyone else? Okay, well, thank you for humoring me, Carol. This is an impossible question that I proposed. And this is because Pseudo-nitzschia can't be identified using light microscopy. The best we can do is actually categorize them into 2 size classes, so either delicatisma, so they're skinny, or seriata, which are larger and thicker. So you actually need molecular techniques like qPCR to distinguish between species. So these are 2 Pseudo-nitzschia species, Australis and fraudulenta. They may look the same, however, genetically they couldn't be more different. So while so on the bottom are sections of nucleotide sequences, and on the top is Australis, and on the bottom is fraudulenta. And while many of their nucleotide sequences overlap, the sections marked in blue highlight differences in the large subunit rRNA region or LSU region, which indicate they are distinct species. So therefore, using genetics allows us to distinguish between species and will allow us to better predict when a toxic bloom will occur or may occur. So early warning forecasting is important for preparing and mitigating the negative impacts of HABs. Currently along the coast of California, we have a model called CHARM, which stands for California Harmful Algae Risk Mapping Project. And to the right is a 3-day forecast of Pseudo-nitzschia abundance on the left and domoic acid production on the right. I won't go into detail, but this is what the output looks like from the model. And although this framework does incorporate a variety of data to make predictions, there are still limitations like predicting at the species level. So therefore, understanding Pseudo-nitzschia dynamics and diversity in our warming oceans is essential. So this leads me to my research question. How does Pseudo-nitzschia growth, community composition, and DA production as a function, change as a function of temperature? And so my first objective was to determine the response of pungens, multi-series, and Australis to different temperatures in batch culture. So my first hypothesis was Pseudo-nitzschia pungens and multi-series will exhibit unique thermal traits, and I broke this up into 2 sub-hypotheses, 1.1. Pseudo-nitzschia strains with higher toxicity will have a higher thermal optimum, and thermal optimum is the temperature where growth is highest. And then 1.2 is DA production will be elevated at higher cultivation temperatures. So then my objective 2 was to conduct laboratory-based co-culture competition experiments at observed temperatures in Monterey Bay, and I'm classifying this at 15 degrees Celsius for the purposes of this. My second hypothesis is that Pseudo-nitzschia species with the highest growth rate at a given experimental temperature will outcompete other Pseudo-nitzschia species. So my entire thesis I've divided into 3 parts. The first part is species identification. We need to collect my species so I can run experiments on them, and we need to identify them. So to the left are the 2 sample sites in Monterey Bay where all of my species were collected, so either in Santa Cruz Wharf or Municipal Wharf 2, and the star represents where we're sitting at the moment. So this is what my collection site typically looks like. And as Holly mentioned in her lovely intro, I do the wharf samplings, so I was looking in the water every Wednesday to see if Pseudo-nitzschia was present. So once I got to my sampling site, I took note of meteorological observations, so water temperature, water color, air temperature, things along those lines. I then performed a phytoplankton net tow and put that sample into a bottle to transport back to Moss Landing in my little electric car. So at the lab, I then looked at my phytoplankton net tow under the microscope to see if Pseudo-nitzschia was even present. So if it was present, I then single cell picked. So that involves single cell picking, washing, and then culturing it until it's nice and healthy. But again, well, I cultured into these 12 well plates. So sometimes you're not always successful. The check marks mean that they grew, and the X means they died. So sometimes they just don't make it. But again, I didn't know what species I was picking, so I needed to identify the cells that were healthy and grew using molecular techniques. And to do this, I sent my sequences out for Sanger sequencing. So by June of 2024, I had all 3 of my Pseudo-nitzschia species, pungens, multi-series, and australis. And all species were collected at the Municipal Wharf 2, except for australis, which was isolated by Kendra, and who was kind enough to share. So all of my isolates were fresh and non-azenic, so basically cultures that are not free from other living organisms. Okay, so now we're diving into part 2, and now that I have my species, I can actually run experiments. So the first series of experiments that I ran were single species experiments. So for my single species experiments, I wanted to determine growth characteristics for pungens, multi-series, and australis at 5 different temperatures, 12, 15, 20, 25, and 30, and I did this in batch culture. I had 3 9-liter carboys in this image, where I grew these single species and measured a bunch of different parameters, which I'll go into further shortly, and I terminated my experiments once I reached stationary phase. So to the right is a video of me performing my daily sampling procedure, where I sampled for photosynthetic health, domoic acid, and growth. And to the left is a schematic of my daily sampling procedure that was beautifully illustrated by Amanda Kanishi in 2024, and whose artwork you will see throughout my presentation. And I'm grateful that she allowed me to use her artwork because I think it's amazing. I also want to thank my amazing REU student, Maya, who's in the bottom of my presentation. She helped me run a lot of experiments at 12 degrees Celsius during her time in the summer of 2023. And so that's what my sampling looked like. I got a lot of steps in those days, which was nice. So now I'm going to go into the different parameters that I measured. So I first sampled for photosynthetic health using a phytoflash fluorometer. And the machine basically detects chlorophyll fluorescence, specifically estimate variable fluorescence, which is FV. And it also calculates maximum fluorescence, FM, and that provides insights into photosystem 1 and photosystem 2. And below is a diagram of that photosynthetic electron transport chain, which you may recognize from your biology class in high school or college, but I won't go into detail on this. And basically, photosynthetic health is important because we want to know if our cells are healthy. And so what is important to know is that an FV over FM greater than 0.5 indicates that the cells are healthy and happy, and a value less than 0.3, the cells are just super stressed out and very unhappy. So the next parameter I measured was particulate domoic acid, which is domoic acid inside or associated with the particle, as well as dissolved domoic acid, which is the domoic acid in the seawater in this case. So to prepare my DA samples, this is me preparing my DA samples at UC Santa Cruz. You might see Rafe in the background or Kendra, but I prepared my DA samples. I performed a methanol extraction, and then I cleaned my samples through a column to make sure it was super pure to go into the LCMS, or liquid chromatography mass spectrometry machine. And I want to give a special shout out to Kendra and Rafe for letting me complete all my DA analyses in their lab and finding space for me and kindly training me. Lastly, I measured growth by fixing my samples and performing daily cell counts. So this graph highlights what a typical growth curve looks like with time on the x-axis and cell concentration on the y-axis. So this graph highlights the 4 typical phases that phytoplankton experience, including lag, exponential, stationary, and death. So the phase that I was most interested in was exponential phase, because these were the values I used to calculate my growth rates for my experiments. So growth rate was calculated using this equation, where A1 and A2 are the average cell concentrations at the start and end of exponential phase, while T1 and T2 are the time in days at the start and end of exponential phase. Okay, so that sums up my single species experiments. And the point of these single species experiments was to determine if and how well a species could grow and produce DA at a given temperature. So before we dive into the results for this section, I do want to lay out what I expect to observe during my single species growth rate experiments as temperature changes. So first, we need to understand the temperature preferences of life. So microbes are classified into 5 groups, as shown in this graph. So we have psychrophiles, psychrotrophs, mesophiles, thermophiles, and hyperthermophiles. And most phytoplankton are in this mesophilic range, so it's about 15 degrees to 45 degrees Celsius. And microbial growth rates are remarkably pretty constant, and so the colder it is, microbe growth will tend to be slower, while at warmer temperatures, it'll be faster. And so above optimal thermal temperatures, your growth rates will decline or decrease. So in my experiments, I expect to see differences in temperature ranges between these different strains within this mesophilic range, which is indicated by the green. So now we're diving into results. So during my single species experiments, I tracked Pseudo-nitzschia species growth at different temperatures. So to orient you to this graph, on the x-axis is time in days, and on the y-axis is cell concentration, which was derived from my cell counts. So each color represents a different temperature, and the top graph is pungens, and the bottom graph is multi-series. So based on my experiments, multi-series will outcompete pungens at temperature extremes that were tested in this. So temperature extremes would be 12 degrees and 30 degrees Celsius. But for instance, at the coldest temperature tested, which was 12, pungens did not grow, allowing multi-series to win out. Then at warmer temperatures tested, both species grew. However, multi-series had a higher cell concentration and faster growth rate. So only at 20 degrees may you notice that pungens did have a slight advantage over multi-series since growth rate was slightly higher, as well as biomass. And then I also tracked photosynthetic health, or FV over FM, which is on the y-axis. So the layout of the graphs are the same as the last slide, with the exception of the y-axis. So photosynthetic health did vary as a function of growth stage, as would be expected, but there was no strong relationship with temperature. So there wasn't a temperature effect. But interestingly, after achieving maximum photosynthetic health during exponential phase, pungens stayed happier and healthier longer than multi-series. So alongside growth and photosynthetic health, I tracked DA production during different growth phases at 12 and 25 degrees Celsius for pungens and multi-series. Pungens is in green and multi-series is in purple. So to orient you, on the x-axis is growth phase, where T0 represents lag phase, TEXP is exponential phase, and TSTA is stationary phase. So on the y-axis is DA concentration. Note the change in Y scales from 12 and 25 degrees. So the first row is dissolved domoic acid, the second row is particulate domoic acid, and the last row, or third row, is total domoic acid. And so as you may have noticed, there are no green bars on any of these graphs, indicating that pungens did not produce DA and is a non-toxic species, unlike multi-series. So you see a lot of purple on this graph. In fact, the toxic species multi-series had TDA concentrations that were more than 20 times higher at 25 degrees Celsius than 12 degrees Celsius. And similarly, multi-series particulate domoic acid, or PDA concentrations, were 150 times higher at 25 degrees than 12 degrees Celsius. So across both temperatures, TDA was highest during stationary phase. Meanwhile, DA levels were lowest during exponential phase. I also tracked DA production at 15 degrees for 2 other species, so Australis, as well as a new strain of pungens, because my other strain died. But that happens, it's biology. So the new strain, same setup, but Australis is in blue and pungens is in yellow. And so, sorry. Yes, so it's in yellow, but multi-series, so if I plotted total DA for pungens and Australis at 15 degrees on the same y-axis as multi-series at 25 degrees, no bars would show up. Although both Australis and pungens, the new strain in yellow, did produce low domoic acid, they were significantly lower, lower DA concentrations than at 25 or even 12 degrees Celsius for multi-series. So based on my experimental data, Pseudo-nitzschia species did exhibit increased ink growth rates and DA production with increasing temperatures. But I still wanted to know what species would outcompete each other if I put them now in the same carbuy with the same nutrient supply when everything is controlled for except isolating the effect of temperature. And so when running batch culture experiments, there are many variables that come into play. However, using a model allows me to assess a single variable at a time. So in order to do this, I had to first determine growth rates at temperatures I didn't test by interpolating my data. So on the x-axis is temperature, and on the y-axis is growth rate in divisions per day. The circles represent my growth rate data from the table above for pungens and multi-series. They're colored accordingly. So at 20 degrees Celsius, pungens had a higher growth rate than multi-series, but this actually flips at 25 degrees. So in order to determine growth rates at temperatures I didn't test, I interpolated my data by fitting a line to my experimental data, and you can see it here. So some nice curves. So now I can figure out what the growth rate for a species at any temperature between 12 and 30 degrees Celsius. So using those interpolated growth rates, I then developed a predictive model to determine which species will outcompete the other based on relative final biomass. So this model applies the Monod model, which describes growth as a function of the amount of nutrients available in the system. So this is the equation that I used, where Umax is the maximum growth rate and are the different values that I actually calculated in my single species experiments and I'm plugging into. S is the amount of nutrients available, and Ks is the substrate concentration. S is the substrate concentration, and Ks is the substrate concentration at which maximum growth rate is half. So with this equation, I can determine what species will win out, and when I have, two strains with identical characteristics and the only difference is temperature. So the strain that is better adapted to warmer temperatures will win out. So as of now, we've been assuming that equal concentrations of each species will be added to the system. When this is the case, there is no temperature impact on growth. And when there's no temperature impact on growth, pungens and multi-series will have an equal chance of winning. So no one has an advantage. But on the x-axis, to orient you, is temperature, and on the y-axis is percent initial biomass for pungens. And so the colored bar to the right displays percent final species biomass. So when it's a teal color, it favors pungens final biomass, and when it's a purple color, it favors multi-series final biomass. In this case, it doesn't really matter if multi-series grows faster at 90% of the relative final biomass because it's skewed to pungens, since pungens will have a head start and use up all those nutrients faster before multi-series can take off. So this is without any temperature impact on growth. However, when you start changing the species proportions being added to this system alongside incorporating the impact that temperature may have an effect, predicting species dominance becomes a little more complicated. So this heat map reveals the trajectory of a bloom developing and how it kind of emerges from a mixed starting community. And so this is under the assumption in a warming future scenario. So the orientation of this graph is the same as the last graph, but now temperature has an effect on growth. So when the percent final biomass of a species is greater than 50%, I consider that that species has now a competitive advantage or will win out. So at temperatures below about 22 degrees Celsius, we don't really need to worry about a toxic bloom, since pungens, which we didn't see produced any domoic acid, we didn't see any green on those graphs originally, is a non-toxic species, so it's favored. However, when temperatures are greater than 22 degrees, we can expect or should expect a toxic multi-series bloom to occur, which is represented by this purple shaded region, by the purple shaded regions. So this model allows me to predict what species will win out by controlling other factors and isolating temperature. So now we're going to dive into the discussion. So identifying the optimal thermal range for Pseudo-nitzschia species growth is important for predicting future bloom events, and this study, like many others, upholds this empirical relationship that's been observed where as temperature increases, growth rate increases, but this is only up to a certain point or limit beyond which growth rate does decrease due to stress. The upper limit for both pungens and multi-series was between 25 and 30 degrees Celsius, which aligns with the thermal range observed in the natural environment. So based on this and other laboratory experiments, it suggests that optimal thermal ranges for Pseudo-nitzschia is between 15 and 27 degrees Celsius, which is within the current and projected sea surface temperatures in the California current system. So on the x-axis here is temperature, and on the y-axis is growth rate. Each line represents a different Pseudo-nitzschia species and across different peer-reviewed papers. So it's three combined peer-reviewed papers on this. So what I'm interested in is at what temperature growth rates are highest. And so you can see from this blue shaded region, you can see that growth rates peak here. And the isolates in this study were higher compared to other studies, but this could be due to a lot of different factors like geographic location, temporal variability, interest-specific variability, nutrient concentrations and light. But despite these different growth rates between this study and others, there was still a positive relationship between temperature and growth. So alongside growth increasing with increasing sea surface temperatures, species thermal ranges may shift. So historically, Australis dominated these massive domoic acid blooms, but with increasing sea surface temperatures, conditions may actually start to favor multi-series to dominate. And this could potentially alter bloom dynamics and ecosystem impacts. So in this image to the right, green plankton from SpongeBob will represent Australis. And in this case, plankton may not like these warmer conditions in their thermal ranges, so the thermal range might shift northward. But as a result, this gives another phytoplankton or Pseudo-nitzschia species the opportunity to come swoop into these warmer conditions, like multi-series, represented by purple plankton. So as previously mentioned, warming conditions may favor multi-series, a toxic producing species, and previous studies alongside this support this positive relationship between DA production and temperature. So the bottom figure has time on the x-axis with DA concentration on the y-axis. So atmospheric PCO2 increases as you move from left to right. There are 3 different temperatures that were tested in this study, and it was 20, 25, and 30 degrees Celsius, represented by different colors. So at 30 degrees, which is that red line, DA production was highest compared to cooler temperatures, especially as PCO2 increases. So the DA concentration detected in this study was lower than what was found in the literature, and this may be due to several factors. One being that usually DA is reported during stationary phase. That's typically when conditions for growing are stressful, like there's nutrient limitation occurring, and so comparing DA levels across the same growth phase is important, as stressed or unhealthy cells typically produce more DA than healthy ones. So in this study, when the cells were stressed, in other words, when FE over FM was less than 0.5, DA concentrations did increase, which is consistent with what other studies have found. So interestingly, in this study, pungens, as I mentioned in my results section, stayed happier and healthier longer after exponential phase compared to multi-series. And this trend became more apparent as temperature increased. So this may be because pungens had an energetic advantage, possibly related to not expending its cellular resources on DA production because, as you recall, pungens didn't produce domoic acid. So nutrient uptake like phosphate and silicate are pretty energetically costly. They're really working for it, and this suggests that the cells diverted energy from DA production back to their growth or into recovery. So even when cells are healthy and high in abundance, this does not determine if the cells will produce domoic acid. So in this study, pungens, as a reminder, did not produce toxins despite being healthy and reaching these high densities. So toxic blooms are driven and controlled by species composition. As you may remember this figure at the beginning of my talk, it's a perfect example of how species composition over cell abundance drives the toxicity of a bloom. So in 2013, Pseudo-nitzschia biomass was 4.6 times greater than in 2015, yet the bloom in 2015 was significantly more toxic. And so this was due to different dominant species. In 2013, the dominant species was fraudulenta, and then in 2015, the dominant species was australis. Australis is kind of the problem child at the moment. But the main takeaways I want you to take away from part 2 is that multi-series is a toxic species and will outcompete pungens at tested temperature extremes, and we may be seeing a shift from historically australis blooms to multi-series blooms in the future. And as our oceans warm, the thermal range for pungens and multi-series may shift. So we may see multi-series more in Monterey Bay, unfortunately. But, and we also now know that DA production is driven by species composition, or basically who's hanging out in the water column. So, and identifying this is important for predicting future toxic blooms. And to go back to my sub-hypotheses, the results from this section supported both of my sub-hypotheses. So as a reminder, 1.1 was Pseudo-nitzschia strains with higher toxicity will have a higher thermal optimum, check. And then 1.2 is DA production will be elevated at higher cultivation temperatures, and we saw this. So tie a neat bow on that. Okay, so then part 3 were my interspecific competition experiments. And this next part of my thesis, I performed competition experiments where I combined species in one flask. And so I created a mixed assemblage and tracked different parameters. But before I created this mixed assemblage with different species, I did run a few preliminary experiments first. And one of these preliminary experiments looked at potential differences in cell size. So I just wanted to know if cell size or biovolume differed between my individual species, because this may impact nutrient uptake kinetics or cell quotas, or its ability to just outcompete each other. I found that australis was 3 times larger in biovolume than pungens and multi-series. And although this was the case, I did not inoculate or adjust the biovolume to make them all equal, because in the natural environment, that's not how it is. You have them naturally, you're not changing their sizes, that's how they naturally occur in the environment. So I proceeded to continue on with my multi-species experiments, but keeping this in the back of my head, knowing this so it can inform the discussion or the results of my thesis. So now I am able to dive into the main portion of my experiment. So for part 3, my experimental design was the same as in part 2, except now each treatment had an equal mixed community of pungens, multi-series, and australis based on cell concentration and not biovolume. Similar to my experimental setup, all of my sampling procedures did remain the same, plus I collected DNA samples and chlorophyll A via filtration. So in order to identify, my individual Pseudo-nitzschia species and track them in this mixed community, I filtered, extracted, and then ran it on a qPCR machine. So qPCR, what is it? It stands for quantitative polymerase chain reaction, and it's a technique used to amplify and simultaneously quantify a targeted DNA molecule, and it detects this based on using a fluorescent signal. So qPCR, think of it like making photocopies, and you're counting how many copies you have in real time. So qPCR allows for real-time measurements as well as quantification ability. So it's pretty advantageous for this portion of the project. So there are several reagents needed for qPCR. So these reagents are combined and then thrown into a thermal cycler that undergoes a series of different cycles involving denaturation, annealing, and extension. So this machine monitors fluorescence throughout this process, which allows us to quantify starting DNA with the help of a TACMan probe. So the fluorescence then tells us how much of that target DNA is present in the sample. So I created a standard curve using serial dilutions of known concentrations of DNA, and to the left is a standard curve with number of cycles on the x-axis and relative fluorescence on the y-axis. And when your sample amplifies, this corresponds to a CQ value, which tells you how much of your starting DNA is in the sample, or you have in that sample. So the CQ values are then plotted against a linear regression plot of known cell concentrations to determine the concentration of your unknown samples. So from the slope of that linear regression, I'm able to calculate my amplification efficiency, and a 100% efficiency means that after each cycle, your DNA is exactly doubling. So this is a linear regression plot for the 3 species I tested. As a reminder, pungens, multiseries, and australis. And I found that all Pseudo-nitzschia qPCR assays, had an efficiency greater than 93%. So that was done using G-block gene fragments. And so on the x-axis is number of log copy number in the reaction, and on the y-axis is threshold cycle, or CQ values. And so a slope of negative 3.4 translates to a 100% efficiency. So in the lower left corner of this graph, you can see that all those values are really close to negative 3.4. So we felt confident with using these assays. But before combining my 3 species, I did run batch culture experiments of each species individually at 15 degrees to determine the feasibility of an initial inocula of about 400 cells per mil, and to see if they'd grow. This is about half of the concentration I used in part 2. So time is on the x-axis and cell concentration is on the y-axis, the first y-axis, and then the second y-axis is FE over FM, or for easier terms, photosynthetic health, or how happy the cells are. So all Pseudo-nitzschia species did grow at 15, with multi-series reaching higher densities than the other species. And all species did achieve were happy and did achieve high photosynthetic health. But although P. australis did not reach high densities, it still had a larger biovolume than the other species, which you want to keep in the back of your mind for our discussion. So I then proceeded, since they all grew, I then proceeded to run my mixed species experiments. So time in days is on the x-axis, and average Pseudo-nitzschia copies per microliter is on the y-axis. So from the raw data, it does appear that australis in blue outcompeted multiseries and pungens. Pungens is in yellow. It really didn't do well. It kind of flatlined, didn't grow. However, once I corrected for the assumption by standardizing the data by copies per cell, this was not the case. So when I standardized all my species by copies per cell, multi-series had a higher concentration. Copies per cell is a way to basically express how much of a genetic target is present in relation to cell number. So the number of copies per cell for multi-series was about 13, while australis was about 135. As I mentioned earlier, australis did have a larger biovolume, which may have allowed it to have more copies per cell. So since pungens died, copies per cell were not calculated for that species. That's why you don't see any yellow on this screen. But it is important to standardize your data because there can be multiple copies per genome. So to figure out how many copies per cell there was for each species, I filtered a known number of cells onto a filter, extracted it, and then ran qPCR and worked backwards. So overall, multi-series dominated over pungens and australis. So we're going into our discussion. So this laboratory study found that at 15 degrees, multi-series outcompeted both australis and pungens in the lab. So this kind of contrasts with what was observed in 2015 in Monterey Bay when we had that massive DA bloom, where australis actually dominated under these silicate-limited and upwell-driven conditions in the bay. So this difference likely reflects the controlled nutrient conditions in the lab where nutrients weren't resupplied, unlike the dynamic, ocean ecosystem out there. So other factors that may have contributed to a difference between this study and others are, one, species with larger biovolumes have a higher maximum nutrient uptake capacity and storage relative to smaller cells. In addition, toxicity can vary depending on, growth stage, and it's important to know what is being reported in different papers. Differences in physiological, genetic, or behavioral traits, among individual strains of the same species can be different. Pseudo-nitzschia species kept in culture for too long can become deformed, like this image. Kind of looks really sad, and produce less toxins over time. So that's just a age of culture effect. It's also important to know that they have their own microbiome, and they're pretty unique. So things behind the scenes could be happening, biology. So although I was able to answer my research question, there are still many knowledge gaps that remain, like calculating and reporting copies per cell. So the table to the right just shows how few papers I've come across or found referencing Pseudo-nitzschia copies per cell. And note, 2 of those entries are actually mine. So there's a lot of scarce information. There's also scarce information on Pungin's growth and toxicity in Monterey Bay. In my study, I found 1 toxic and 1 non-toxic strain. So that was kind of fun. So more research needs to be done there. Pseudo-nitzschia genes for toxicity were only recently identified, but more research is needed to understand, if these genes turn on and if they're upregulated. This is a very active field of research at the moment. If people, are interested, I would say pursue that. We still do need to understand Pseudo-nitzschia community composition, not only as temperature changes, but also other things change, like the acidity of the ocean and nutrient inputs under our rapidly changing climate. So the main takeaways from part 3 are, 1, to my knowledge, this is the first study to use qPCR assays, Pseudo-nitzschia qPCR assays, to track Pseudo-nitzschia species composition in a laboratory setting. Also, at 15 degrees, we can expect multi-series to dominate over Australis and Pungens. And 3, it's important to acknowledge that there are many factors that can influence species composition. So now we're going to go back to our hypothesis 2. I'm claiming that it was inconclusive. So although all species grew individually at 400 cells per mil and were, physiologically happy and healthy, the variability among biological replicates were pretty high, limiting my confidence in my calculated growth rates for that portion of it. So this variability may be explained by a combination of factors, like working with older strains and reducing the initial cell densities. So this is a stepping stone for future research, and I would definitely try and complete this again under different temperatures as well. So in conclusion, what should you take away from all of these slides? Well, 1, as sea surface temperatures warm, these conditions may favor multi-series, a high DA producing species, to outcompete other Pseudo-nitzschia species like Australis and Pungens, and temperature does play a pretty critical role for predicting species-specific toxic Pseudo-nitzschia blooms. So with that, I want to thank my amazing committee for their support. Thank you, Holly, for always having my back and my best interests at heart. You've definitely made me a better scientist, and I am extremely grateful for that. Sarah, for your online, thank you for your support and challenging me to think outside the box. Tom, thank you for your positivity and patience, teaching me Python. And Rafe, thank you for your support and guidance and always, willingness to help, especially when it came to running my DA samples and interpreting those results with me. I want to thank all of my scholarships and grants that funded this work, especially my molecular work. Molecular work is sneaky expensive, so super grateful for that. And I want to thank the Moss Landing community as a whole. I definitely couldn't have done it without you all. And my cohort especially. I love my cohort. They have been super supportive. I want to thank my RU student, Maya Clark, for being amazing and helping me run some of these experiments at colder temperatures. I want to thank my only lab mate and roommate, well, ex-roommate, Olivia Pollock. She is actually driving cross country, so she couldn't be here today, but she has been a great support system. I want to thank the Marine Pollutions Lab for taking me in and giving me a job and still inviting me to their barbecues, like monthly gatherings, and just supporting me throughout this process. I want to thank Amanda for always her positive attitude, and as well as helping me, teaching me how to use this light microscope that she had, as well as Image J. That was new to me. I want to thank Steve Cunningham for, when my lab shut down, I had a really hard time, and he took me in under his wing, and so I was still able to make progress on my research as well as make money. I would like to thank the Moss Landing Marine Labs facilities team. They're a great bunch. I want to thank Billy especially for coming in late at night when I had accidents in the lab, and he was grocery shopping and came and helped me. I want to thank Kelsey Montalto. She has been such a support system and a statistics guru, so if anyone needs help, I would definitely contact her. I want to thank Jonah for always being positive and more than willing to show me how the microscope in the ichthyology lab works. Thanks, Jonah. I'd like to thank Noah for taking the time to take pictures of SEM for my phytoplankton species. Andrew Kim for teaching me how to use a light meter, a cute handheld light meter. I want to thank my collaborators, Kendra, phenomenal woman, for letting me use her species and training me and just being so helpful. Stephanie for molecular work, Amanda for her beautiful illustrations that you saw throughout my presentation. I want to thank my family and friends, my mom, who's my biggest supporter, and she's sitting in the front row. Thank you so much for coming and supporting me, and in general. Thank you, Liz, for coming. It's been, I just love my family. Thank you to the McGrath family, everyone that supported me on this journey, especially during this difficult month. Difficult month with someone passing recently, and all of the love and support that I've received. I also want to thank my East Coast friends and my boyfriend, Josh. He's the one that made the beautiful bread. Has been very, very supportive. My Bigelow friends that are keeping me on the marine science track, and my friend Hannah, who's listening in on the East Coast. Thank you to my previous mentors, Dr. Pete Countway and Dr. Cindy Heil and Sarah Class, that encouraged me to even go to grad school and contact Holly Bowers. So that was the best decision I could have made. So I'll leave you with that, and here are some additional photos. And I welcome any questions, comments, or concerns at this time. Yeah, Carol? Oh, sorry. I can also bring it.
Good job, Hannah. That was great. I have two questions. I'll keep them short, though. I'm really interested in the methods you use for qPCR and how you standardized each sample from like day to day. Did you do more about it? Or how did you keep them consistent, I guess? Did you have to like cell count each one to see like what was in there, or how did you standardize each sample for qPCR?
Yeah, to standardize it, I worked with the qPCR assay efficiencies a lot, so that's why getting to that 93% or greater was really important, because we want to make sure when it doubles, it's almost doubling 100% that DNA. I would run several serial dilutions in the morning, fresh ones. I'd make fresh ones every 24 hours to make sure it was consistent throughout the process, so there wasn't any deviation or things going wonky.
Thank you. And then my next question was the DA slide in like your first section.
Mm-hmm.
How did you, I don't know much, how do you calculate the total DA versus like particulate DA? Was it like your entire experiment, or was it?
Yeah, that's a great question. To calculate, let's see how difficult this is to go back. Okay, so here to calculate my DA, so it's broken up into 3 parts. there is some behind-the-scenes work that goes on, but for total domoic acid, I'm basically combining my particulate and dissolved and adding them to get my total. Yes, they are in different units, but I put them all in nanograms per mil in order to combine them. There is certain math that goes behind it. Rafe is a great resource, my committee member who ran most of my samples for that. There's a Sachs factor that for calibrating. There's different things that go into it, but that's a general gist. We can talk separately.
Thank you. Good job once again.
Good job, Hannah. Congratulations for finishing. I had a question about your temperature treatments. So I was curious what the relevance of them were, especially for the 25, which is a temperature that we don't typically see, at least in this area, the Monterey Bay. And so I think you had 12, 15, and 25, and so I'm wondering how you got there and why you use those temperatures for your experiment.
Yeah, that's a great question. So as I previously mentioned, I go down the wharf every Wednesday, take air temperature, water temperature, so that's what my 12 and 15 are based off of. And then my 25 and 30 degree temperatures were based off of what other papers have done. So what's common in the phytoplankton literature, and I wanted to amplify it, so I really shot up the temperature to be able to see those temperature effects in growth, DA concentrations, as well as biomass. Yeah.
Thank you.
Hey, Hannah, wonderful job. What an amazing amount of work that you did. It's incredible. I was curious actually about this slide. So you said that pungens was, you found out it was non-toxic. So did you know beforehand that pungens was a non-toxic species, or like did you find out during this experiment?
I found out during this experiment, but that's the thing with species. You never, like pungens, sometimes it's toxic. It's typically less toxic than other species. So yeah, you don't see any green in this, but at 15 degrees, a different strain of pungens did produce small amounts of DA. So I didn't know really going in.
Cool, thank you.
Great job, Hannah. That was so good. I just have a question about the multi-series. So I know in Southern California, DA is pretty prevalent. Has there been any studies looking at the species composition down there and what species is driving DA's prevalence, if it's like multi-species that does better under warmer temperatures?
Yeah, there are fewer studies on diversity of Pseudo-nitzschia species. There have been a couple down south. So there was one that used long-term records to basically look at the different species that were there or like that have been recorded. But typically down there, it's still been Australis, yeah, that dominates during those blooms.
Thank you.
Yeah.
That was really great. Thank you. So I was curious, how much DA, like what concentration of DA do you need in the environment to start seeing toxic effects? And how much, well, what kind of quantity of Pseudo-nitzschia do you need to start seeing that amount of DA?
Yeah, so the DA that I detected in my experiments, like in during the 2015 bloom, like it was 100 times more than what was observed in the lab. So around like that, you start seeing effects. You have to get out of the, femtogram kind of category and start getting into like whole numbers. But again, in culture, DA production's a little different than like what you would observe outside. But yeah, 100 times more, you start seeing effects like in the 2015 bloom.
Got it, thank you.
Hi.
Okay.
I was just so impressed with the cultures where you had all three species together, and multi-series just really dominated. And then you mentioned that in the blob, Australis was dominant in 2015.
Yes.
Do you have any like hypotheses as to why, you know, in your cultures, Australis didn't outcompete them, but during a bloom, it might?
Yeah, that's a great question. That's a really good one. I think it's a lot of different factors. Because I would feed my cultures about once every week, and my multi-series, once it was isolated, was pretty robust, looked great, but I was given Australis, and then over time, it started looking deformed. I think it might have been age of culture effect. I also think it depends on strain variability. I think there's just so much research that still needs to be done in order to figure that out, and like having higher just getting more isolates of the same species. Because I collected all of my species in 2024, by 2024. So it could have been affected by different environmental factors going on outside, whether I collected in the summer versus the winter time. So season also impacts, you know, what they're going to act up, how they're going to act. Yeah. Great question, Monet.
Thank you.
Hey, Hannah. Do you think, I don't know if you've covered this, but did the rate of temperature change really impact your communities as much? Like when you did your different temperatures, how did that impact it? Like did you time it, or was it a constant increase?
Yeah, if I'm answering your question right, I did acclimate all of my cultures at least 10 days between temperatures, and acclimating stepwise, so it wouldn't be a shock to them. So I wouldn't see, it wouldn't affect the results of that one temperature. If that's what you're asking.
Yeah, I was wondering if a shock would kill them off, or
a shock would, because I learned that the hard way.
Oh, okay.
It does. If you don't do a stepwise, so I originally went from 15 to 25, and a 10-degree jump was that they were not happy. So, yeah, definitely, using stepwise is great.
Cool, that's cool.
Yeah. I don't know what's next. I'm not in charge. I don't know what's next.
Sorry, falling down on duties. All right, another round for Hannah. And I didn't hear some of these stories like, oh, I tried to go 10 degrees and it died. I'm just now learning that.
I didn't tell that.
So now the committee and Hannah, we whisk her away to the whale room, I think, or think tank or somewhere. And then we ask her some questions, and then she's free to go, and we do some paperwork. And so please help yourself. I think I bought too much coffee, maybe. I don't drink coffee, so I have no idea what I was doing. But please, whatever is extra, take it to the student lounge, and it'll get you through to Kelsey's defense later on. So.